Hello, everyone. So today we will discuss and extend our discussion as we were discussing about the regulation of enzymes. So in my previous lecture, I discussed with you that what are the different factors or with different substances which can work as a regulatory molecules for the enzyme activity. So we have seen the activators and inhibitors. So our today discussion is mainly focusing on the inhibition because there is the more extensive types and the details which we need to talk about the enzyme inhibition. So before discussing that what are the different enzyme inhibitors, so there are some of the questions which we need to go through. The first thing is why. So why enzyme inhibition required? Does it has any advantage or significance? So yes, so to prevent excessive function within cells and organism. So that enzyme inhibition is not only in a case that synthetically we wanted to inhibit some enzyme activity. This is some of our natural procedures, how our living cells our different metabolic activities are conducted. So the nature is also managing the enzyme activity through the inhibition and sometimes through the activators also. So usually when a simple example is when there is some of the cancer cells and they are multiplying a lot. So there is a need that what is the enzymatic function which is responsible for their multiplication and the reproduction. So that enzyme activity has to inhibit, that has to stop, that has to cease. So for this, we need a substance or a molecule or a ligand which can stop that enzyme activity, and these are called inhibitors. Now, the how portion of enzyme or the protein blocks access to enzyme active sites. So this happened through the pocket or the cleft, which we call the active site of an enzyme, which is responsible for the enzymatic activity. So that kind of protein maybe or so any receptor. So if there is a substance which can bind fit to that enzyme active site and it is blocking the excess of enzyme to the normal molecule which needs to bind with that enzyme for particular enzyme activity. So here are some of the examples we will discuss here. So trypsinogen has extra piece of protein gene to cover active site and keep it inactive. So when there is a need that it has to stop the trypsinogen activity, so what it do that this will block that active site. Pancreatic trypsin inhibitor ensures that any trace of active trypsin are inactivated when not necessary. So obviously, when our metabolic activities are conducted, so some of the amount, for example, if we are taking any of the protein and that has to digest, so it has completely digested. So the what I type of hormones or the secretion or enzymes has to release for conducting that activity. So now if there is no food, if there is no material for digestion, so definitely that has to stop, because otherwise these secretion can damage or living structure. So we are all made up of the protein largely, so definitely then they will be damaging for us. So for example, one of the example is ulcer. So what is happening that when there is a more secretion of these uh, digestive secretions, and that is getting to the very acidic one, so that is damaging your stomach layer, that's the protein layer, and then that is causing some kind of inflammation, and it gets gets very worse. So that is the advantage and the significance that why the inhibition of enzyme activity is necessary and what is the importance of that. Now we will see that enzyme inhibition uh, can be divided into the two different inhibitory processes, or you can say phenomenon. So the enzyme inhibitions um, could be the reversible, and that can be irreversible. So now I need your attention because there is the minor difference and a technical difference which you need to understand to cover the comparison or difference between the reversible inhibitors and irreversible inhibitors. So as the name states, so you're familiar with the reversible reactions in chemistry and the irreversible reactions in the chemistry. So when we are showing any of the reaction with the double half headed arrows, so it means that reaction is moving forward, and in the same ratio it is moving backward, right? So this is simultaneously moving from forward and backward. So anytime if we control the conditions, so we can go for the one major side of that reaction. So reversible inhibitors, basically they are binded to the enzyme through the non-covalent bonds. So because they are through the temporary interactions, so that can cleave easily. So dilution of enzyme inhibitor complex result in dissociation of the reversibly bounded inhibitor and recovery of enzyme activity. So what does it mean? That once we needed some of the enzyme to work on a substrate and it is going through the different steps, and this is the induced fit model, that there is the enzyme substrate complex, then enzyme product complex, and then eventually there will be the product released to the system and we will get the enzyme originally. And whenever there is a need of that activity and we need that, we have that starting material on which that enzyme has to do some of the reaction, so that enzyme will come again and bind to that substrate and eventually we will get the product. Now here the enzyme is releasing and it is separating from the product. But what happens in the irreversible inhibition? So let's say, and one important thing is that this irreversible inhibition can work on enzyme um, inhibitory complex, right? And or that can be in a initially enzyme and substrate complex, right? Either that can inhibit at the level when there is an enzyme and this is not working with the substrate at all. So irreversible inhibitors can work on any of these two states. Either they can capture that enzyme substrate complex, or either they can bind to the enzyme, so an enzyme will not be able to conduct its activity on the substrate one. So now here you can see that at the end, we are not having that enzyme and only the product, which is the straight doubly headed arrow. So this is not the reversible reaction, this is irreversible reaction. So when the product has formed, it's fine. And here this has inhibited one, and now the enzyme has poison and this is not able to conduct its activity again. So we will see what are the different inhibitory molecules and what are the different kinds of inhibition. So very first of this is the competitive inhibition. So an inhibitor, let's say if we just focus on this diagram, this is an enzyme. Here there is a substrate which naturally has to go and fit with that enzyme to produce certain product. No, if we have in a system, we have an inhibitor, right, which is not allowing that enzyme and it doesn't like that that enzyme has to work on that substrate. So what it can do is, so in case of the competitive inhibition, that inhibitor will show up itself in a more promising way, that it will fit to the active site of the enzyme, and now the substrate is not having any access and there's no chance for the substrate to attach on that active site because that active site has already been filled by the inhibitor. Now, why we call this competitive inhibition? The competitive inhibition is that kind of inhibition in which the inhibitor fight or compete with the substrate for that active site attachment or the binding, and then substrate is losing its way to the enzyme. Uh, so, and if we just plot that curve in terms of the competitive inhibition, so that would be rate of the reaction, substrate concentration. So here, one thing you have to focus is that if we are just going through the normal enzyme reaction. So there is a peak and then that gets straight, and we know that why it gets straight, because when all of the active sites will occupied, so that velocity or Vmax or reaction rate is not exceeding or it is not enhancing any further. So no inhibitor present, and if we have added an inhibitor, so what is happening that this enzyme activity is not getting that high, right? It is slowly going through some kind of enzyme activity or they totally stop, but we can reverse that inhibition if we increase the substrate concentration a large. So we can resume enzyme activity if we are supplying too much of that substrate concentration, so that will gain its Vmax level. But obviously that is the more time taking, and this is the slow one as there was a rapid increase in the Vmax. Now the next is the non-competitive inhibitor or inhibition. So non-competitive inhibition are when there is an inhibitor molecule and this is not having that courage to face the front or the active site of an enzyme, but it to attack from backside any other site other than the active site, and it bind with that enzyme. So as a result, now the enzyme's active site, which was suitable with the shape of that substrate molecule, is no more compatible for that substrate binding, right? So that kind of inhibition, because now the enzyme activity has inhibited, so that inhibition is the non-competitive inhibition. And when we plot this non-competitive inhibition, this is giving you the idea that in case of the non-competitive inhibition, whatever we do, so even if we increase a lot of substrate concentration, so even then that enzyme will not achieve its highest point, which was the Vmax. So it will always be at the lower V level. So it is not giving you the higher or the maximum reaction rate, what was its normal enzyme activity that it was able to convert the starting 100 substrate into the product one. So now that will be reduced a lot. To understand that concept of competitive and non-competitive inhibition, one of my most uh, uh, famous or my favorite example, which I used to give to my students, is that you just compare an organization. So and in your daily life you have such kind of people. So one is that if you are performing very well and you are getting the very good appraisals and your boss is appreciating you, but eventually there is a person who wanted to be at that place. So there are the two ways to replace you or to lower you, or maybe to just get at least your position is. So one thing is that that person do that much hard work, or he or she is showing um, best of them, and then eventually due to their hard work and their smart work, they are getting the better place. So they are competing with their skill, with their knowledge, or with their efficiency. So that is the competitive inhibition. So they are stopping your appraisal or your appreciation, because now that appreciation and appraisal has shifted to that person who was in competition with you. Now the other type, which is the best form of the non-competitive inhibition, is that sometimes um, people are not that skillful, and um, they are not able to work or perform well, but they don't want it to see you uh, in a progress and see you at a high level. So what they will do is that they will create the hurdle in your way. So they may poison your boss, and they use any of the non-competitive and illegal mean to poison your boss against you, whatever, maybe just backbiting, maybe just portraying you negatively, that whatever you're doing is just a time scoring and you're not performing it, but you're showing off, etc., etc. So that is that the person don't have the courage. That person is not able to perform, but that person is poisoning your boss. So whatever you will do, if your boss wants get poisoned, so you will not get that appraisal. So that is the non-competitive inhibition. Now we'll move forward for the next kind of inhibition. Uh, Before this, we just can compare the difference between the competitive and non-competitive. Though I have showed you in each of that slide that in case of the competitive inhibitor, you can do this, that if you increase the substrate concentration, even then, if there is an inhibitor, the enzyme can get its maximum level. So this is something that if you are having that competitor who is working hard, so you start more strategically and you work more smartly. So maybe you resume your position. But in case of the non-competitive, when that person is not in front and he or she is doing something you're not aware of it, so you can't compete overseas, so you can't plan accordingly. So then that even by increasing the substrate concentration, that enzyme will not reach its Vmax level, right? So the difference is, and these are very important and usually the mostly asked questions, and obviously whenever you are going through the medicine chemistry or maybe the pharmaceutical, so you should know that how um, what is the difference and how we can cope with the competitive inhibitor or the non-competitive inhibitors. So effect on the major difference is the effect on Vmax, which is the maximum velocity or the velocity of the reaction. So non-competitive inhibitor cannot be overcome by increasing the concentration of substrate. Thus, non-competitive inhibitors decrease the Vmax of the reaction. Now, the effect of a competitive inhibitor can be reversed by increasing substrate concentration. As sufficiently high substrate concentration, the reaction velocity beats the Vmax observed in the absence of the inhibitor. So, competitive inhibitor case, we have the same Vx as in the non-inhibiting reaction, but different Km value, rate of reaction, right? So for the non-competitive inhibitor, the Km is the same but different Vmax, because now this is not reaching its maximum velocity reaction, the maximum capacity an enzyme can conduct on the substrate. Now, another important inhibition type is allosteric inhibition. So this is somehow just the kind of non-competitive inhibition, but it is why we started this separately, because this is having more, you can say, the uh, uh, specialized sites which are other than the active sites. So they are enzymes which have some of the compartment where the inhibitors can fit in, right? So allosteric regulation can be through the allosteric activators also, and that can be through the allosteric inhibitors also. So right now we are discussing allosteric inhibition, and obviously for the activator means which we have discussed in our regulation of enzyme. So allosteric compound, if it bounds and it is making the enzyme active site more compatible for the substrate, so that will be the allosteric activation. But right now, just look at the allosteric inhibition. So let's say this is the enzyme shape. This is its active site, and this is the allosteric site. So allosteric site is any of the site or the position or the pocket which is not the catalytic site, but where any of other substances can attach, right? So this is allosteric site. Now the inhibitor, like the non-competitive inhibition, this is not binding to the active site, but this has a more compatibility, and that is a more proper shape to fit in with that allosteric site. So now through that allosteric attachment, what is happening to the enzyme? If you just look at this, so its shape has deformed, right? Now this is not able to bind with this with that enzyme. So it means that enzyme activity has ceased and stopped. Another important terminology which is also you need to know, though this is not a part of our syllabus, but you should know that what are the allosteric enzymes. So allosteric inhibition is any of the inhibitor which is binding or attaching, and it is reducing or stopping the enzyme activity by the attachment at another position which is not the active site, not the actual catalytic position, but it binds and it deforms the enzyme active site, so that is it is not able to bind with the substrate and to conduct that enzyme activity. So allosteric enzymes typically have multiple active sites located on different protein subunits. So now the next one is a feedback inhibition. So you know that whenever we go for the any professional job, and even if you're working in any of the industry, so the feedback is very important. So the feedback could be in term of to improvement or capacity building, and that feedback is and especially if you just apply this on the industrial batch. Let's say if you are visiting any of the industry which is creating some of the product, let's say if that is the water or the drinking water industry, so there is the first thing is the filtration and the mineral addition of the water molecule, then that fill in the PET bottles. So if there is a huge batch of the PET bottles which is already filled, and the counter is overcrowded, and now the next step is their labeling, right? So what would be done for that batch is that they have to stop the filling, and they have to stop uh, that belt where there is a large number of the bottles which are just waiting for their labeling. So that is a feedback. So that feedback has sent to the back side on that batch or the production area, that just stop filling further PET bottles, let's complete their labeling. That is a case in the biological reactions and the activities that most of our metabolic reactions. So what is happening that once that is an enzyme, it binds with the substrate, it conducted one of its activity, and that was the intermediate substrate, or you can say the first product, and then this product has to be the part of another reaction. Like for example, in our body system, the glucose is converting into the acetyl CoA, and then acetyl CoA is entering in another metabolic cycle. So that is the one product could be the starting material of another reaction. So let's say now the enzyme 2 is working on that one, and intermediate substrate is there, and now the enzyme third is there, and this is the end product, right? So what is a feedback inhibition is that this is end product. Now this has produced in a larger amount, and we don't need that further to produce. So it means that reaction has to stop from the starting site, because if that will be produced, definitely that if that compound is present in the system, so definitely its enzyme will come to work on that enzyme. So therefore, that is necessary that this chain has broken there or inhibited there. So this product, if it is exceeding its required amount, so that feedback will be sent to the system or the metabolic cycle, and this end product will bind to the allosteric site of that enzyme, and in a way that it is sending that signal in a higher concentration of that end product. So now this enzyme is no more able to work on that substrate. So that substrate will be saved or stored as a bulk, like a glycogen is one of the example that extra sugar, carbohydrates are stored in our body and animals as a glycogen. And whenever we require or system require, then there is the cycle which use that, and it consume and regenerate the carbon dioxide in the water and the ATP when our body require that glucose to break up for generating the energy, because our physical activities are dependent on the ATP. So ATP is the or cellular currency or metabolic currency, you can say. This is the energy carrier molecule, and it is an allosteric inhibitor of some of the enzymes involved in cellular respiration, like the glycolysis, a process that make ATP to power cellular reactions. So when there is a surplus amount of energy, so that will bind to the enzyme, and this is not conducting any further reaction on that sugar molecules to generate the ATP, because obviously our system is managing in a that way. Whenever we are doing extra exercise, so our body start generating glucose, and when we are at the very lower pace and we are not moving, and then this is not moving, gluconeogenesis or that is not happening, because now there is no further need to decompose or break down the glucose into the carbon dioxide and ATP, because right now we don't need any extra energy. Now, another thing which you may not get in most of our medicinal chemistry or the biochemistry books, because this is the most emerging in the recent strategy for regulating the enzyme activities. This is the enzyme degradation, and this is the only kind of you can say the inhibition in which is a destructive one, because there is the protein molecule which is damaging. So what does happening, so when we talk about enzyme degradation, you will get that term PROTAC. So PROTAC, if you just Google, there are a lot of papers you may get, because now from these few years, 5-6 years, or maybe the less than that, the people have started to work on the PROTAC and the protein degradations. So this PROTAC is the abbreviation of Proteolysis-targeting chimeras. So these are some of the synthesized molecules, or these are molecules which our body can also synthesize, that this is responsible for degrading some of the proteins. So what is do is the ability to selectively degrade protein with bifunctional small molecules is protein degradation. This has like this is a molecule. This is one kind of degrader, and they are having the bifunctionalities. So they have the number of different functionalities from where they are making the two sides. So the one side will bind with the active site of enzyme. Another side will bind with that ligand, right, or the receptor where it has to work. And there is a protein, because every enzyme is associated with some kind of proteins, or maybe you can say the holoenzyme, which has a protein portion, and there is a cofactors. So these proteins are required. Their attachment with that enzyme are required for their proper functioning. So enzymes are protein in nature, so there may be some kind of embedded proteins which are responsible for conducting or helping that enzyme to for the enzyme activity. That could be the part of that enzyme chain. So that is some of the part of that enzyme can be responsible, or that could be shown as a P. P is showing the protein. So now this is the heterobifunctional ligand. It has two sides, and usually they call as these are the linkers. So what does it do is that firstly this just get bind with the ligand and the enzyme, and now what is happening to this is that what was that protein is with that enzyme, so this just has removed from that enzyme, and now this inhibitor is just dissociating that protein from the enzyme, and we just get the enzyme with no activity. So this is called the protein degradation, and the next slide will make your concept more easy. And this is the difference between the inhibitor and the degrader. As I said, that the degrader are basically the more destructive, not in a negative terminology. This is basically whenever we need so inhibition make an not effective. So where the inhibition is not effective, the degradation is the more effective therapeutic strategy, because then it can dissociate and it can cleave, it can degrade that protein which is responsible for that enzyme activity. So this is the active target, this is the inhibitor molecule, and when that inhibition was there, so the turns to be inactive target, and now there is no enzyme activity. Now what degraders do is that this is a degrader molecule and this is the active target, so it will just degrade that protein. This protein is not in that original structure that it can conduct any um, enzyme activity, and this degrader is just resumed from the system. So this is in some kind of the cases when the inhibition is not effective, and inhibit can also affect other regular activities also. So only that specialized protein kind of degraders are synthesized, which can only specifically target that protein structure and degrade that one. So this was all about the inhibition, enzyme inhibition, their types, how they do work. And so now we have completed the enzyme uh, topic, and I hope you get that concept. And if you have any question, so just do write on YouTube, just under the lecture videos, so I could answer you and just clear your queries and if you have any confusion. Thank you so much.